right. We're talking today with Dr. Adela Oliva Chavez, who's a professor of entomology at Texas A&M University. And we're going in depth today, and her research focuses on interactions between three things: ticks that spread disease, sometimes called the vector, and bacteria carried by the ticks that cause the disease, and then their hosts. So those could be humans or other animals. And we're going to try to understand a little bit more about this. And then from what I understand, this research also can have applications in the area of medicine, even vaccines, right? And veterinary treatment for, things like livestock, if I'm not mistaken about that. So Dr. Chavez, welcome to the Insect Shield blog. It's really nice to have you here.
Hi, thank you for having me. This is a great opportunity, and I really love that you're educating people about transmission of vector-borne diseases in general.
Yeah, this is so cool. I think we have other entomologists that we work with, and they're kind of focused on the prevention side. Doing things like one of them, Thomas Mather, is working on a kind of a sign that can be placed with a QR code, so that can be scanned and people can get information immediately on their phone. One of his things is like when they're out, like going to go for a hike, they can't really change their clothes at that point. Yeah. I mean, they need something they can use right then. So that's the kind of stuff we're doing. But today, we're really kind of honored to have you here and talking about the research, more in depth into this area. So I think with those three things, just the ticks themselves and the pathogen, that they carry, and then the human sort of, that's going to suffer from these infections, I guess, is what we're talking about. So, and you've done a recent paper about this too, I think.
Yeah, so what we look at is we look at the interface between the three. So if you think about it, when they are transmitting those pathogens is when they are biting you. So that is what we call the interface at the skin site. And it's not a big area, it's just a couple millimeters around where the tick is biting. And what happens is that ticks have evolved to actually diminish the immune responses in animals, invertebrates, and also in humans. Because they are what we call obligatory blood feeders, meaning that it's not like a mosquito, for example. The mosquito, you have the male that it doesn't bite humans, it just feeds on sugar. Ticks, both the female, the male, and all the life stages, they need to feed on blood, because that's where they get the proteins and the fats in order to pass to their next life stage or to develop the eggs. And they feed for a very long time. So if you think about the bite of a mosquito, it comes for a couple seconds, it pierces through the skin and it finds a blood vessel, and that's how they feed. In the case of the tick, what they do is they are what we call pool feeders. They come with their mouth parts that we call a hypostome, and they have what we call a chelicera, that is like two cutting extractors that go like this.
Right.
And they cut through the skin, and the tick puts the hypostome in. Okay. And in the first 24 hours, they put a solution that we call cement, that is like a glue. And people actually, it's funny that you mentioned the medical application, because the people are looking at this cement as a probable glue for surgeries, because it's very potent. Like if you pull a tick, it's really hard to get it out.
Right.
And it's because of this cement cone that they put. And it also protects the mouth part from cells that might come to try to kill the tick. And after those 24 hours, they start injecting other molecules that delay the closure of that wound, and also work as vasodilators, making the blood vessels get bigger, so there is more blood coming in, so that they can feed better. And all those molecules are also helping the pathogens, because there you have an area that is going to have diminished immune responses, and it's perfect for any pathogen to actually get and establish there, because you have a different set of cells that will not normally be there. So they can escape your immune response, and then that's how you get Lyme disease and other diseases. Because as I was telling you, Ixodes scapularis transmit many more pathogens, not just Lyme disease. There is human granulocytic anaplasmosis, there is ehrlichiosis in the area of Minnesota and Wisconsin, and there are other pathogens that are transmitted by the same tick.
Okay. Yeah, that's something that people haven't been aware of, but some of these are more serious, right, in terms of the diseases that they can spread.
Yeah, because what happens is that the people can get even more sick. So there have been studies where they have shown that when people have Lyme disease and another disease called human babesiosis, this is a protozoa, so it's actually similar to the agent of malaria.
Oh, wow.
Yeah, so it's actually an eukaryote. It's not a bacteria.
Okay.
And people get very sick with that particular pathogen.
Yeah.
And the other one that I work with is Anaplasma phagocytophilum, and causes human granulocytic anaplasmosis. And that one makes it hard to diagnose. So when people come in, they are misdiagnosed for Lyme disease, and then you get the late Lyme disease. That is when people start developing the palsy, the arthritis, and then the heart problems. Yeah, because if you treat all these diseases early on, it's not such a big problem. Like human granulocytic anaplasmosis just causes like high fevers and you feel really sick, but it's like flu. But if you're immunocompromised, you can even die.
Right.
And in the case of Lyme disease, if you don't treat it early on, there are chances that you're going to have complications.
Yeah, yeah. So this is really interesting in terms of the way that these bacteria, I guess, have evolved, right? Because they have this sort of ability, but the tick is kind of facilitating the same kind of thing. So I think I read that they have kind of a symbiotic relationship. Yeah.
Yeah, they do, because so we don't think about like the amount of time that they have been in a relationship. So for example, Borrelia, the first Borrelia-like organism found in ticks was 15,000 million years ago. So they have been together for millions and millions of years ago. It's just that we either have not been aware that they cause disease in humans, or we have not been paying attention. It's like what you said, many of the times is the fact that people are not aware of the pathogen being in a specific area and like a specific set of symptoms. We tend to say, this is something else. Like my sister, when she was younger, she got rickettsia, and the doctor said, oh, this is dengue, because they are so used to diagnosing dengue back in where I am from, that they just misdiagnose.
This is dengue is the disease that you're referring to? That they
dengue fever. Yeah, I come from Honduras, and we have like a very pronounced dengue season. But my sister actually got a rickettsial disease that is transmitted by a different tick. It's also tick transmitted. But back in those days, we didn't know about it. So they were like, oh, this is dengue, because she had fevers. But she got treated with antibiotics and then she got better. But that's one of the problems. We're not aware of the presence of the disease, and then it gets misdiagnosed. And so because they are symbionts on the tick, they have also evolved a way to change the saliva of the tick. So the pathogen can also manipulate the expression of specific genes in their advantage. For example, Borrelia burgdorferi needs a particular protein that is called SALT15. And it's very interesting because what the bacteria does is it coats itself with this protein that is in the saliva of the tick. And this protects the bacteria from antibody recognition.
Okay.
And the bacteria itself increases the expression of this protein in the saliva of the tick.
Wow.
Yeah, so these little creatures have evolved all these mechanisms to be able to infect you.
Right, right. Yeah, I mean, like you say, then I'm sure, we're only just now kind of learning about how these things work. So it's, in some ways, the more you look into it, the scarier it gets. I'm sure that's not really the point. But yeah, so that's really fascinating. Now, if you're doing this research, obviously you want to do more. I think you point that out in your paper, like we're only kind of scratching the surface in terms of our understanding. But what about the applications for this? Like, I think you mentioned, possible development of vaccine would also be based on the research, right?
Yeah, so the paper that I published, it actually looks at something that is some small particles that they are called exosomes and extracellular vesicles. And we all produce them. Like we humans, we produce exosomes. And what we find is that in the saliva of ticks, we find these microvesicles, these exosomes, and that many of these exosomes already, they have all these effector proteins that the pathogens are taking advantage of.
Okay.
So if we can take these exosomes, which they already have all these proteins that the tick needs and the pathogen needs, and we can then use them as vaccine, inject them into an animal or inject it into humans after FDA approval and all that, then the humans will, and the animals will develop an immune response against those proteins. So if we are recognizing these proteins and we are destroying these proteins, then we can stop the tick from feeding, because now we can kill the tick. And now we can potentially also kill the pathogen. So that's one of the things that we're investigating right now. Can we use these exosomes as vaccines to prevent tick feeding? And what proteins in these particles, in the exosomes and the extracellular vesicles, are being recognized by the immune responses? So that then, instead of, because the process of purifying these vesicles is very complicated, the idea that we have is if we can recognize what proteins they are, then we can engineer our own vesicles. So if you think about and most people don't know this, but if you think about the COVID vaccine, this COVID vaccine is messenger RNA that is inside of what we call a liposome. This liposome is actually a vesicle, an artificial vesicle made to carry these vesicles, these messenger RNA. That's what we want to do. We want to make our own liposomes carrying these proteins so that we can vaccinate people.
Right. That makes sense. And so you're talking about the extracellular vesicle, right? That's the key. Okay. But every organism in some sense uses those same kinds of vesicles, to transport or interact between cells.
So yep, they do, but each of the cells in your body makes their own vesicles. So the message in the vesicles in your body are different to the messages in the tick. And even so these vesicles are fascinating, because now we're finding that they can be used for many things. So they are using them for cancer diagnostics, for looking to see if you can use it for diagnosis of Alzheimer's, even malaria, because any disease changes the cargo of the vesicles, the message that is being sent. Like in the case of cancer, the cancer cells change the vesicles so that they can metastasize. It's like sending a message to wherever they are gonna go. Okay,
Right.
we're going to establish in this other organ, so we're sending all these vesicles. So
Oh,
now
wow.
we can harness those vesicles, knowing that they have a different message, and looking at, okay, what is this different message, and what in these vesicles is causing an immune response, so that we can take advantage of that immune response in order to then attack whatever disease, the disease or a pathogen, or in my case, the tick.
Right, right. Okay, got it. So the vesicle is just this thing that communicates from a cell. You can have that communication happening inside the cell as well. Is that right? Okay.
Yeah, and
yeah.
The interesting thing is that like these vesicles accumulate in certain like liquids. So for example, you can find certain vesicles in the saliva. And so that's what we're working with, the vesicles from the tick saliva. And I actually am also working with the vesicles from the midgut of the tick, because
From the gut.
the gut, yeah, because if we can produce antibodies that will damage the midgut of the tick will die as they are feeding. That's the hypothesis that we have. So all your organs, all the cells in your body, they are producing these messages. So we are looking at that. How can we use these messages that the tick is using to leave in order to kill it?
Right. So what would that look like in terms of administering that? Is that called a vaccine then when you apply that knowledge? Okay.
Yeah, that it is. It will be an extracellular vesicle-based vaccine.
Okay, got it. And so, I mean, there's talk about this because I think the idea right now is there's no vaccine for Lyme disease, right?
Yeah, no, not for human use. So there was one back in the 90s, I think. It was before I came to the United States. That was based on a protein that is in the outer surface of the bacteria.
The surface of the bacteria?
Yeah, and it's a protein that is needed for the bacteria to infect. But what happened is that there was a public backlash against the vaccine, because some people were actually reacting badly to the vaccine. So that was taken out of the market. And now there is a vaccine available for dogs that is based on chimera. So what happened with this protein is that this outer surface protein is that it changes from different isolates. So that was the other problem. The vaccines against one particular bacterial isolate will not work against another bacterial isolate. So nowadays they are making what is called a chimera, that is a protein made of several proteins. So that the same vaccine will work with all different isolates of the bacteria, but this is only available for dogs. So there is not anything that is available for humans. From what I know, they are trying to see if this vaccine is safe for humans, but it's not available on the market.
Is that, I mean, obviously it has to go through the whole approval process and so forth, so that could take years.
It can take years.
Yeah. And you know, Insect Shield works with a number of Lyme organizations, and they're kind of across the gamut of, you know, prevention and treatment, diagnosis. You know, there's still, like you were saying, there's not great understanding at this point, so you can go a long time without getting a proper diagnosis. But is the vaccine, I mean, who would want to use the vaccine then if it were available for humans? Would it just be everybody that gets, you know?
Yeah, I will actually, because it used to be the Lyme disease was affecting specific groups of people, like people that tend to be outdoorsy, people that work in parks, things like that. Like those
Right.
were more prone to have it. But right now, like I was telling you, because of the expansion of the tick populations, and the fact that now we have urbanization, like suburban areas going into wooded areas, so that you have
Suburban,
deer
like
passing through
a
houses.
backyard or something has up
Yeah,
against the woods.
out
Yeah.
in the woods, exactly. So nowadays, people can be exposed to a tick carrying the pathogen in their backyards.
Yeah.
And that's a problem. So I would say that anybody that is in areas where there is high prevalence of Lyme disease should get vaccinated, because I know of people that had the disease and it's very debilitating. Like people that have palsy and lose movement of one region of their body, and or have developed like heart problems and cannot do the things that they were doing before. Yeah.
So along those lines, I mean, these bacteria are somehow benefiting, right? I mean, they're in the body to benefit from the environment, or is it just that they can breed in there, or what? Why are they doing this? You know,
well, it's like anything, they survival, right? Because so these are a special kind of bacteria. It's what so these bacteria, they need to be in both the tick and the vertebrate host, because there's a process that we call transovarial transmission that happens in some pathogens. That is when the mother can pass it to the offspring in the case of the vector. So let's say the female tick passes it to the larvae. That doesn't happen with Borrelia. That doesn't happen with Anaplasma phagocytophilum. They are not passed from the mother to the offspring. So if you don't have that vertebrate host that gets sick, then the bacteria is dead. They disappear. Yeah, so they are doing it to complete their cycle. They are selfish. They are not. They don't care. I'm gonna make this person sick. I just want to survive and whatever.
Right. Oh, I got it. So one of the folks that we interviewed, Dan Wolf, was talking about this, that it's the case because it's not passed from one tick to another, that the larvae at the very early stage don't carry it so much. So you could be bitten by those larvae, right? But you're not going to get Lyme because they don't have it yet.
They don't have it, yeah. So if you are, that's what I tell my students all the time. If you're bitten by the larvae, don't worry about it, because they are not likely to have it. They might have viruses like the Powassan virus, because that can be transovarially transmitted.
Powassan?
Yeah.
Which is also very can even be deadly, right?
Yeah, it's like the tick-borne encephalitis of the United States. And you find it in certain areas, like in New York area, in the Northeast mostly. We find cases. It's not that many cases, but those are going up too.
Right. So I think it's important for people to understand those different, because we're talking basically about the deer tick, right? That carries Lyme.
All of those. Yep.
Yeah. But there's like 4 different stages of the life cycle. Is that right? Or so? Okay. Yeah, so we're talking about larva, nymph, but the nymph, from what another of the people that we interviewed, Logan McCullough, he had nymph bites, and like he said, like 20 nymph bites, but that was enough to sort of carry this severe disease. So it's, you know, larvae are not a big deal, but the nymph, and then after that, they grow bigger, most people are only familiar with the large, whatever the adult.
The adult, yeah. And the funny thing is that the adult is not so much of a problem as the nymph, like what Dr. Logan said. So most of the transmission happens during the bite of the nymph, because they are so small and you can have many and you don't realize it.
Right.
So what happens during the cycle is that usually you have the eggs, that's the first stage, the eggs hatch, and then you have larvae, and the larvae will feed on likely an uninfected host, but they can feed on a mouse, or like a white-footed mouse, or another host that is infected with Lyme disease. Then they acquire the bacteria. Once they acquire the bacteria, they will go through the molting. So they will retain this bacteria, and then the nymph is infected. And then when this nymph bites a human, then they will transmit the disease. And the more ticks you have transmitting it, because they do these, so ticks have pheromones that they send to each other. So it's like a chemical, a cue that they send to each other. I found a place where we can feed. So they congregate, and they feed together. So you like have like a party, yeah. They are telling you, the drinks here, come over.
They're not social distancing either when they have
these
parties.
They go like, oh, we don't care about Corona. So you can have like sometimes ticks aggregate, and then these immunocompromised sites is even more immunocompromised, because you have two ticks throwing these effector molecules that are affecting our immune system, and then you have even more bacteria that establish, and then you get even more severe Lyme disease.
Right. So that was one point that Logan McCullough made, which I found really interesting. And he said, the first kind of line of defense is really the immune system. So if it's already compromised in some way, then you're much more susceptible to bacteria. But that's not like a guarantee. I mean, anybody with, even if their immune system is in good shape, is still susceptible to the bacteria.
We are all susceptible to the bacteria. We all have so when bacteria use what is called receptors, they use receptors that are found in bodies to bind and to infect us. And we all have them. Just some people have a better immune system, and they might be able to clear the disease without it becoming severe. But even if you are not immunocompromised, you might have just like some other disease that is affecting your immune system, and then you might get severe complications. So it's not that many people that develop delayed severe Lyme disease, but it's a big percentage, like 10% of people might actually, and not just immunocompromised.
So it sounds like, one of the keys there, like you mentioned, is really early diagnosis, or finding these ticks, early on. And that's something that people can do. I mean, from our standpoint, insect shield, it's like prevention is much easier than this other, reacting to actually getting the infection. So we really recommend, I mean, it's not hard to put on, insect
repellent
clothing, and then to follow kind of guidelines of like walk in the center of the trail and don't be traipsing through. But if it's in your backyard, I mean, that's tough too. And then the dog also can go out and kind of
bring them over. But that is where it becomes key checking yourself. Like even when you are taking a shower, if after you went outside to work in your garden, if you were close to the wooded areas, because what happens is that it takes quite some time for transmission to occur. Like I was saying, the secretion of the saliva, it changes through phases, and it's the same with the transmission of pathogens. So in the case of bacterial pathogens, like the Lyme disease agent, this pathogen has to replicate inside of the tick. So what happens is when the tick bites you and it starts to get in the blood in, that sends a cue to the bacteria telling them, oh, okay, that now we can be transmitted. So they start replicating in the midgut, and then they have to make their way to the salivary glands. So for Lyme disease to occur, that tick has to be feeding for more than 48 hours. So if you got that tick in the first 24 hours when they were feeding, you are likely to be okay.
Yeah.
So people should be checking themselves after doing outdoors activities or when they are taking a shower, like we shower every day, right? So if you live close to wooded areas where your dog might bring a tick, check your kids when you're showering them, and check yourself when you take a shower.
Yeah. Yeah, and so the other important part of that, I guess, is if you find a tick, the way to remove it is also really key, because you don't want to like squeeze it or do that, try to do it with your fingers or, you know.
Don't put Vaseline. I have heard everything, like put Vaseline on top of the tick. That doesn't
work.
Right.
Well, I mean, it's killing the tick is not the goal. You know, it's like you can be mad at the tick, but it's not really going to help. So I mean, it's like important to use these very fine tweezers and then grasp it. And like you said, it's got that kind of cement. So you really have to be careful to pull it like straight up. Those kinds of things. So that's really critical too. You know, and that's like Dr. Mather, like I mentioned, you know, it's like education around this, you know, the tick habitats or, you know, all this stuff, and then removal. That's really important. And I think there's still a lot of kind of just not a lot of understanding out there, you know, about these things. So.
Yeah, and people don't know where to find the information that they need. That's also part of the problem. But that, because of the internet, is becoming a little bit easier. So you can actually put on YouTube how to remove a tick, and you're likely to find a video showing you, or like, for example, what you guys are doing at Insect Shield, you are doing these education videos. So it has become a little better because of the internet. And like the CDC has a webpage where you can see how to remove a tick, and they give you like a list of things that you can use as repellents that work against tick, that there is like scientific data showing this works. So I think it's becoming a little better. We just have to let people know this information is out there. You can find it.
Yeah, and I think the one downside of just trying to use Google is a lot of times there's a lot of information, but it's like, well, what's the real, you know, story? So we're trying to do that too, to kind of make it easier to find actual stuff. And then, you know, there are more support groups, I think, than there used to be, and the internet has helped that. You know, people can find doctors or organizations that help them to find doctors, and you know, can help support people that have these longer term illnesses as a result. So this is all great. I really appreciate you taking the time to talk to us about this, and it's pretty fascinating. I especially like the idea that, you know, by understanding this and doing more research, we can come up with better ways to solve the problem. And I know, you know, people get very excited when they hear there might be a vaccine, but I don't think they really understand all the, you know, important things that are involved in getting that done.
So. Yeah, because there are many aspects to it. First, we have to identify what will work, what will generate an immune response. After that, we have to investigate, is this safe? If we inject it into people, are we going to make them sicker? Or is this going to actually, instead of blocking the transmission, is it actually going to make it worse? And then you have to do the same thing with kids, because the immune system of kids is different to a well-developed immune system of an adult.
Oh, okay.
So those are all the things that had to go through the process. So that's why it takes normally several years for development of a vaccine.
Right.
But the good thing is that the understanding the process is not, we're not limited to just vaccines. We might find molecules that if we treat somebody with this specific molecule, we enhance their immune system, and then their immune system can kill the bacteria. Okay. So there are like prophylactics too. Yeah. Like you can take a pill or like get an injection of something that might prevent you from acquiring when you go. Like that's what they do with malaria. There is a prophylactic for malaria that if you go to a country where there is malaria, you can take it before going there.
Right.
So there are other options that people are exploring as well, besides just vaccine development.
Right. Okay. Got it. Well, Dr. Chavez, it's been a pleasure here. Thank you so much for making the trip from Texas. And we'd love to have you on again at some point if you're interested. And I hope your continued research goes well, and you can keep finding these insights about pathogens, and especially Lyme.
Yeah.
We like to try to keep ahead of that and educate our readers and viewers. So thanks so much. That's Dr. Adela Chavez from Texas A&M University Department of Entomology.
Thank you very much. It was a pleasure.